I will introduce our second to last speaker, Pratiksha Thakore. Pratiksha did her graduate work in Charlie Gersbach's group at Duke, where she developed CRISPR-Cas9 tools for epigenome editing. And she's now a postdoc in Aviv Regev's lab at the Broad, combining her passion for genome engineering with high content single cell profiling to understand both autoimmunity and cancer. And the title of her talk today is High Content Single Cell Screens for Unraveling Gene Regulation Mechanisms in Cancer Immune Evasion. So take it away, Pratiksha.
All right, first I'd like to thank everyone for the opportunity to speak here today and be part of I'm really honored to be part of a lineup of exciting speakers, and I'm happy to share our recent work developing Perturb-CITE-seq in order to understand mechanisms of cancer immune evasion. So it's a really exciting time for the field of functional genomics. New technologies have really transformed the way we study gene regulation in biological function and disease. And early in my postdoc, a wise immunologist once said, every disease is an immunological disease. So in turn, it's a really exciting time in immunology too. First, with technologies like CRISPR-Cas9, we can now make genetic perturbations at will and in a massively parallel manner. Genetic screens are really powerful ways to search for gene function and understand cell circuitry. Traditionally, these screens are performed by delivering a pool of perturbations at about one guide, if we're talking about CRISPR per cell, to your biological model, and then you can do some type of selection or enrichment step for a phenotype of interest, something like cell survival or some type of flow-based marker. And then you can count the barcode to your population and use enrichment to find regulators of your phenotype. So these types of screens have been really high throughput and really powerful, but they introduce some key challenges. We must know what the readout will be ahead of time. We must do this via the selection step. And because this type of readout is usually based off of a marker of some sort or cell survival, it's only really amenable to asking certain types of biological questions or measuring specific types of phenotypes. So in 2016, our group and others developed a screening method called Perturb-CITE that addresses this challenge head on. We start off like a normal CRISPR screen, delivering a pooled perturbation library, but in Perturb-CITE, we can measure the effects using single-cell RNA-seq. With this single-cell output, we can both match the cell to the perturbation it received and the transcriptome, which provides a rich source of information to both measure the impact of the perturbation of the gene on gene regulation and cell function. So this has been a really exciting advancement for CRISPR screens, and it generates a powerful platform to ask questions about cell fate and to inform biological discovery. These high content readouts can really open up us to asking new and complex questions. But to date, Perturb-CITE screens have only been done with a transcriptional readout. So this can be pretty powerful, but we're interested in asking questions that would be also interesting to have not only RNA, but protein-based readout. And you can imagine in the field of immunology, a lot of the phenotypes that we're interested are also, we're interested in studying involve not only transcriptional changes, but protein changes as well. So we first became interested in extending Perturb-CITE beyond transcriptional readouts alone to also include protein when we were playing the screen to investigate how cancer cells escape T-cell killing. Previous work by our group had identified an immune evasion gene program expressed by patient melanoma cells, and this program was predictive of cells that can escape immune surveillance and survive immunotherapy. So this program essentially has two modules. Cancer cells survive by shutting off various cell-cell communication programs, antigen presentation, and immune signaling. So they essentially are hiding from the immune system. And they, at the same time, turn on cell cycle and ribosomal protein genes. So we wanted our screen to take advantage of this transcriptional program that we have and have really high content readout. And we noted that our transcriptional program includes many key immune evasion markers that are not only important transcriptionally, but are also cell surface proteins that are accessible by antibodies. So in order to maximally read out this function, we wanted our screen to not only include transcriptional readouts, but also cell surface marker expression. So to do this, we turned to a method called CITE-seq developed at New York Genome Center. And in CITE-seq, you can tag your cells with barcoded antibodies. And these antibodies are barcoded in such a way that they can be incorporated into existing droplet-based single-cell RNA workflows. And then so you can both get a measure of protein levels for your antibodies of choice and RNA at single cell resolution simultaneously. So we incorporated these protein readouts into Perturb-CITE, to an extended Perturb-CITE. We took this workflow and collected matched transcriptome, protein, and guide RNA information and developed a computational method as well to interpret all of these types of data. And the study was performed as a collaboration with Benny Zar's group at Columbia, and it was really fun to work with a really brilliant set of co-authors, or collaborators, Chris, Johannes, and Katie. And it took a lot of different types of expertise to bring this study together. So having both protein and RNA helped improve our gene detection. So immediately we can look at single-cell RNA-seq of key immune evasion markers. And one of the things that you immediately notice is that a lot of the genes that we may be interested in or that we know are important to immune evasion have very sparse signal in single-cell RNA-seq or high dropout rates. But if we look at protein from the same cells, we see less dropout and more robust signal. And with CITE-seq, we can multiplex many different antibody targets, and the number of targets is ever increasing for performing these types of CITE-seq assays. So, so immediately we see that including these protein readouts can have a lot of value or add a lot of value to our detection power in our screens. So in order to test Perturb-CITE-seq, we turned to a great patient melanoma model developed in Benny Zar's group. So they had patient-derived melanoma cells that express Cas9 and T cells collected from the same tumor, so matched T cells. We set up a Perturb-CITE-seq screen targeting 248 genes from the program in patient melanoma cells. And we had a couple of different contexts to look at, with and without interferon gamma treatment, and after interferon gamma treatment and with T cell co-culture. So this beautiful UMAP on the right shows how this experimentally setup really kind of yields a flower, 3 different distinct contexts for us to dissect and study these gene regulatory mechanisms. But today I'm going to focus on our results from melanoma cells that particularly survive T cell killing in co-culture. So the first thing we can do is we can turn to kind of the traditional bulk CRISPR screening method and look at CRISPR guide enrichment and see which perturbations yield improved survival of our melanoma cells to T cell killing. And then so when we look at our guide RNAs or perturbations that are enriched in cells that survive, we immediately see genes that regulate the JAK-STAT pathway and interferon gamma response. Interferon gamma response is important to T cell activation, so this made total sense to us. Disrupting these pathways would, of course, be important to improving survival to T cell killing. So this gave us confidence in our screening setup and the results that we were getting. We also see other interesting genes pop up, like CD47 and CD58, that confer survival melanoma cells to T cell killing, and we'll come back to some of these later. So we use that enrichment data in order to identify potential drivers, or regulators of these cancer cells' ability to escape immune evasion or escape immune surveillance. And now we could turn to our information-rich RNA and protein profiles to delve into how these perturbations are actually affecting gene programs. So shown here is a regulatory matrix that we developed and derived from a linear model in order to understand the impacts of our perturbations on gene and protein expression. So the rows here are RNA and protein features that were measured in our single cell data. And the columns are guide RNAs, are targeting specific genes for knockout. And you can see these organized into modules that act on gene expression the same way, so perturbations that act in similar manners, and then gene programs that get regulated in similar ways. So first we can zoom back in on our JAK-STAT perturbations that I had mentioned earlier, and you see that these perturbations act very strongly and in a coherent manner to affect gene expression in our model. One thing I forgot to mention in the previous slide is that the color mapping tells us about how the guide affects gene expression. And then so what we see here is that interfering JAK-STAT perturbations, repress many genes involved in antigen presentation machinery, a lot of chemokines associated with anti-tumor immunity and inflammation, interferon response genes as expected, and checkpoint genes. So these are well-known players in immune evasion, and so that gave us a lot of confidence in our model. And then that allowed us to return to kind of the larger data set and map, including the full perturbation set from our screen. So as I mentioned before, we've organized our perturbations into co-functional modules and then matched them to co-regulated gene programs that they affect. This provides a really rich source for probing gene regulation mechanisms and kind of looking through and generating new hypotheses about what might be happening in immune evasion. One interesting that we noted is that shown in the boxes in our target modules are hits from the CRISPR enrichment analysis that we did that are important to survival of melanoma cells from T-cell killing. So we know that all these genes are impacting the survival phenotype in the same way, but the actual gene programs and the mechanisms by which they do this are distinct in some cases and converging in others. So this really highlights kind of the extra layer and the kind of deeper you're able to go in understanding mechanisms when you have this kind of high content screening readouts. Today, I'll talk a little bit in more detail about one of the hits that we were interested in studying, CD58, which was both enriched in our T-cell killing analysis or survival of melanoma cells to T-cell killing, and also included in our Perturb-CITE screen. So if we look at CD58, it's a cell surface protein that's found on a lot of antigen-presenting cells and lots of other cell types. It can engage with CD2, which is found on T cells and natural killer cells. We had it as an antibody that we targeted in our CITE-seq data. And what we can see is that CD58 expression is depleted in cells after co-culture. And this effect is independent of whether or not the cells are treated with interferon gamma. Consistent with this, we took a look at JAK-STAT pathways and saw how they may be affecting CD58 versus other interferon gamma signaling genes. So our JAK-STAT perturbations, as we had mentioned before, had a really robust effect on different interferon gamma signaling pathways, but actually did not have knocking out or interfering with JAK-STAT did not negatively impact CD58 expression. And then so I just wanted to highlight again that if we compare our protein signal versus our RNA for CD58, and as well as other interesting immune evasion genes like PD-L1, we see that we really get a more robust picture of what's happening what's happening to these genes and proteins when we deliver different perturbations. We really need that protein readout, which is more robust in our model than the single-cell RNA-seq readout that we're able to achieve. So once again, the Perturb-CITE-seq readout was really adding value to our ability to understand what these perturbations are doing. So we're interested in understanding if CD58 might be a mechanism, might be acting on cancer immune evasion in a mechanism that's independent to JAK-STAT signaling, as kind of indicated by our analysis so far. So what we next looked at is the CD58 gene regulatory program and how that's impacted by different perturbations. So each point in this chart is a guide RNA perturbation. And we can see that knocking out CD58 has a strong impact on the CD58 regulatory program. But interestingly enough, the JAK-STAT pathway actually has opposing effects. Knocking out the JAK-STAT pathway actually has opposing effects on the CD58 regulatory program. So once again, this indicates that these are independent mechanisms of regulating immune evasion. And then so knowing that these two screen hits have different mechanisms, or potential different mechanisms, can really be informative and useful information if we're thinking about next steps and generating hypotheses that would be important, for example, to designing therapy. You could have a hypothesis that a combination treatment targeting two different pathways would yield interesting and exciting results for immunotherapy resistance. So CD58 has been implicated in immunoevasion previously. We validated this in our model by doing competitive assay for T-cell killing. We found that if we have CD58 knockout, we get improved cell survival to T-cell killing. And the CD58 knockout did not impact MHC1 or MHC2 expression. And we could look back at our previous transcriptional data from immunotherapy-resistant patients, and we see that CD58 expression is reduced in immunotherapy-resistant patients. And this actually also is a good opportunity to highlight that CD58 is actually a gene that does not have a homologue or does not have a corollary gene in mouse. So it was really a gene that we could only investigate by having this really exciting patient melanoma and matched T-cell model. So in summary, we showed how multimodal screens can improve detection of cell surface markers, and having both RNA and protein allows us to really better interrogate immune evasion mechanisms and some of the functional ways that this occur. And then since, of course, this is an immunology symposium, it'd be completely right to ask, but what's happening in the T cells, right? So we're well aware and really excited about the idea that a lot of interesting phenotypes in immune cells, such as T cell exhaustion and viral infection or cancer, have been marked and well studied by their cell surface protein expression. So we're really excited about the potential for Perturb-CITE-seq screens in immune cells and are actively pursuing this. And so to close, I'd like to return to my earlier thoughts about how it's a really exciting time to apply functional genomics immunology. We just kind of showed one flavor of a Perturb-seq screen with RNA and protein readouts. It's just kind of one possibility we've drawn from a really ever-growing Perturb-seq toolkit that we've been working on at Aviv's lab and other people have been developing in the field. So kind of any type of perturbation that you can barcode, and there's lots of different advances in high content readouts that we can combine and use this to continue to ask really outstanding and interesting questions in cell fate and function immunology, in different tissue contexts and interactions, and then harnessing this platform to design exciting immune therapies. And then I'd also just like to once again thank all the wonderful people who worked with me on this story. Really, Chris, Katie, and Johannes, we all together put together a lot of interesting, really critical biological, technological, and computational expertise to make this study happen. And Aviv has been an amazing mentor throughout this experience. And thank you all for listening today. Thank you.
Thank you very much, Pratiksha. Great talk. If anyone has questions, please type them in while I'm talking and I can read them out. But I was going to start with something that you brought up very briefly at sort of, I guess, in the future directions. I was wondering whether you had done any combinations with chromatin assays with ATAC-seq or anything, and whether there was better or worse correlation between that and some of your other readouts compared to the RNA versus protein.
Yeah, so combining RNA and ATAC for Perturb-seq readouts is a really active area right now. So Saima, who's another person in our lab, has just come out with a nice technology to get both of these types of readouts. And then also we've been working with the Smybert lab to get protein and ATAC readouts. And there's interesting information that you can get from the ATAC, such as transcription factor motif analysis if you're looking for drivers that wouldn't be available in RNA or protein, obviously. And then so we're really excited about applying those, but I haven't looked at this particular model yet on ATAC and RNA together.
Great talk. You talked about you gave us some nice examples of how you could get more data from the protein and in places where there was dropout of RNA-seq and stuff. Do you ever find evidence or see hints that there's actually discordance, that you see regulation at one level of the transcript, but you're actually picking up something that's happening post-transcriptionally that you can then detect at the protein level?
Yeah, so we think this is a really exciting question to look into for especially in the way that perturbations might differentially impact the RNA versus protein. So we didn't see a lot of examples of that in this particular study. I mean, we had a pretty small panel of antibodies here, but we're doing some larger scale Perturb-CITE-seq screens now, and that's one of the questions that we're looking at in our analysis.